Vision video, Mr. Phillips, and today we are going to be looking at C2.1 chemical signaling. So what we're going to be looking at today is the chemical signals and different types that are in the body, and then we'll be looking at how receptors work. So you need to be familiar with different chemical signals that are available, and we'll be talking about these in different videos in more length. So the neurotransmitters obviously links to nerves, cytokines often links to immune response, calcium ions are used within muscles and within the synapse, and hormones is what we're going to focus on today. So you need to know some of the key things, what main molecules make them up. You need to know what mechanism of transport they use. So do they use the blood? Do they diffuse across a synaptic gap? What distance do they travel? How long they last? And what their effect might be. So you can pause the video here and have a look at this if you want, but I'm going to move on. So when we're talking about signaling chemicals, the word that we should really be using is the idea of something called a ligand. A ligand is a chemical messenger. It attaches to a receptor, and that then triggers some sort of response within the cell. It will always be a complementary shape when it attaches to the protein receptor, and it causes a change in that protein receptor that stimulates then the response in the cell. By the end of this video, we'll have a look at some of those in detail. Ligands are molecules that then bind selectively to a specific site on another molecule, and a ligand binding site is the receptor or an enzyme which it binds to. And the two must always be complementary in shape. So we need to compare the difference between enzymes and receptors. We're not really looking at enzymes in this topic, but you should hopefully know that an enzyme is a substrate and an active site coming together. So when that happens, the substrate is converted to a product and then is released. The binding is very brief, the enzyme is unchanged, and this happens repeatedly over and over again. With chemicals and receptin binding, the ligand binds to the receptor site and doesn't act as a catalyst, so it might remain and bind for a long period of time. It might form a conformational change of the receptor, and eventually the chemical will be released unchanged. So examples that we need to be aware of are neurotransmitters. And neurotransmitters are chemicals that transmit signals across a synapse, about 20 nanometers in gap. And you need to watch the neurotransmitter and synapse video to study this. Cytokines, as I said, will be covered more in the disease topic. They're small signaling molecules, and they may affect nearby cells, or they may affect in a more systematic approach and affect the whole body. They bind to receptors as per usual on the cell surface membrane, and one cytokine can bind to many types of receptors, so it can affect many different cell types, as so triggering the immune response. And calcium ions are used, as hopefully you know, to bind to troponin, triggering the sliding filament theory in muscle contractions, and they enter the synaptic presynaptic bulb, triggering the release of vesicles with neurotransmitters in the synapse. So we're going to focus on hormones, and hormones here you can see are chemical signals that are released from an endocrine gland. They go into the bloodstream and then attach to a receptor on a target gland, causing a specific response. They can affect long distances, so for example from your pituitary gland to your ovaries for FSH, and can have a long-lasting effect. These are the endocrine glands you have down here. Many of these we study at different points in the course. So how would you define a hormone? I'd define it as a chemical messenger produced by an endocrine gland and transporting in the blood to a target organ where it attaches to a receptor cell and activates an enzyme in the cell. Now, most of the examples we're going to talk about are protein hormones, which have to attach to that receptor on the outside of the cell, but lipid-soluble hormones can diffuse straight through the cell and can bind with a protein receptor inside the cytoplasm. We'll look at these later on. So the endocrine system where all these hormones are being released is controlled by the hypothalamus, and the hypothalamus links the nervous system to the endocrine system via the pituitary gland. So the hypothalamus will receive signals and it sends messages to the pituitary gland to release different hormones. The hypothalamus has specialized areas called nuclei, each operating different control centers that it receives information from. So here you can see the nuclei, the different groups around the hypothalamus. and that might include areas that monitor blood temperature, glucose control, osmolarity of the blood, hormone concentration, sense organs, other parts of the brain. And here you can see a bit more detail if you want to pause and have a look at the different hypothalamus nuclei. So the hypothalamus is then connected to the pituitary gland via a narrow stalk, so you can see it here, okay, and it's controlled by nuclei in the hypothalamus. So different nuclei activate the different bits of the hypothalamus to release different hormones. The hypothalamus then triggers either the pituitary or, sorry, the anterior or posterior areas of the pituitary gland, which are two different lobes releasing different hormones. These hormones you will talk about elsewhere in the course, so I'm not going to spend too long on these. Again, you can pause the video if you would like, but the posterior pituitary gland releases antidiuretic hormone, which you'll be familiar with from the kidney topic, which controls the selective reabsorption of water in the collecting duct of the kidney. Oxytocin is the hormone that is responsible for starting contractions and the positive feedback of contractions at the end of child's labour, so to create a childbirth. The anterior pituitary gland releases a series of hormones, and again, if you link it to the ones you know earlier in the course, so thyroid stimulating hormone you might not have heard of, or the middle one here, but you will have heard of follicle stimulating hormone and luteinizing hormone from the reproduction section of the course. You also need to be familiar with growth hormone, which increases the rate of mitosis, increases amino acid transport into cells, rates of protein synthesis, and uses fats for energy. So when we need to start looking at the types of receptors, it is important to know that there are different types of receptors in the cell. So in cells where the lipid molecules, lipid hormones can get through into the cell, we have intracellular receptors. So these are receptors, protein receptors that are found inside the cell cytoplasm. So the signaling chemical, for example, testosterone, which is a steroid hormone made of lipids, so it can pass through the cell membrane, will go into the cytoplasm and bind to receptors. These will have hydrophilic amino acids, so they remain undissolved in the cytoplasm, so they're not hydrophobic. So they remain dissolved, in the cytoplasm, so they can interact with the liquids in the cytoplasm. We then also have transmembrane receptors, where the signaling chemical does not pass through, and the chemical will attach, as you can see here, on the outside of the cell membrane, and that causes a conformational change of the protein, which goes into the membrane and into the cell, which then normally activates a different enzyme on the inside of the cell. And we'll look at the example of adrenaline doing this shortly. These have hydrophobic amino acids on the surface. Okay, so this area here would be hydrophobic, and they have hydrophilic layers at each end to attach, to attract, and to allow them to interact with different pieces to aqueous solutions. Sorry, my words got trapped in there. So how does this work? Well, if you've got the intracellular proteins, then estrogen is an example of a lipid soluble protein, lipid soluble molecule that can go through. It goes straight through the cell membrane. So in this case, the estrogen molecule can pass through the membrane and to a receptor on the inside of the cell in the cytoplasm. So this receptor will have hydrophilic amino acids, so it can remain dissolved in the solution and it will interact with the solution. This then, as you can see, changes or causes a change in the shape of this, which will then trigger a series of enzyme control reactions, altering DNA expression in the cell. So there you go. So binding of the signaling molecule creates an active ligand receptor complex that activates this protein, and that will regulate gene expression in the cell, causing the inhibition or promotion of transcription of particular genes, which is called epigenetics. Receptors, transmembrane receptors, the signaling chemical, are normally a hormone that's non-steroid, so it's a protein hormone such as adrenaline, cannot pass through the plasma membrane, so it has to bind to a protein receptor on the cell surface membrane. The protein receptors have hydrophobic amino acids on the surface, so they're attracted to the A polar tails of the phospholipids in the bilayer, so it allows them to stay in the bilayer. What then happens is the ligand binds. It will change the structure of the protein, the binding receptor protein, we should say, on the inner side of the membrane, and that then becomes catalytically activated. So what it normally does then is it activates an enzyme like this one, and then that then causes a secondary messenger inside the cell, which conveys a message to go into the cell. And we'll look at a specific example. So the example we're going to look at is adrenaline. Adrenaline is a water-soluble hormone secreted by the adrenal glands, as you know, hopefully for the flight or fight response. And effectively, we need to know a little bit about adrenaline and how it works. So adrenaline is also called epinephrine, and it binds to adrenergic receptors on the plasma membrane and triggers a response to gain more oxygen and glucose for vigorous activity. So adrenaline will help glycogen break down to glucose in the muscle and liver cells. It causes your airways to dilate, allowing more oxygen in for happy respiration, increase your ventilation rate, speeds up the heart rate, and it allows arterial muscles to the liver to widen, and arterial muscles to the gut to constrict, reducing flow. So this increases the rate of ability for you to do activity and exercise by increasing the rate of respiration. It has lots of effects on different organs, so you can pause here and have a look at this bit if you would like to make more information on it. So how does adrenaline work? Well, there's a series of reactions that you need to know. So the adrenaline attaches to the protein receptor. Remember, adrenaline cannot go through the membrane because it is a protein, not a lipid-soluble molecule, and it attaches to the receptor protein, which will have a complementary shape to. That then causes a conformational change in shape of the receptor protein, which will then change the shape of it inside the membrane, and that causes a series of reactions which activates an enzyme called adenyl cyclase. That enzyme then is activated, which triggers ATP to be converted to cyclic AMP, and that activates an enzyme called protein kinase enzyme, which converts glucose and hydrolyzes it down, glycogen into glucose. That glucose then can be released into the bloodstream and used in respiration. So that is your key thing. So here, this would be adrenaline, and it would be what we call the first messenger. They cannot enter the cell. The same for glucagon, and which has this same effect. And they require a secondary messenger inside the cell. In this case, it's called cyclic AMP, which activates enzymes inside the cell. So you need to know there's a first messenger and a secondary messenger when it is protein hormones. And the example that you need to know for those two is glucagon and adrenaline. Again, glucagon would have the same impact as adrenaline here. So let's look at it in more detail. How does it actually activate the enzyme? So adrenaline binds to the specific receptor site because it's got a specific complementary shape. Here you can see the protein is the receptor here. This activates an enzyme called a G protein. The G protein then activates the enzyme adenyl cyclase. Adenyl cyclase or adenylate cyclase converts ATP into cyclic AMP. And then cyclic AMP activates an enzyme called protein kinase A. Protein kinase A actually activates then phosphorylase kinase, and then glycogen phosphorylase is activated, which converts the breakdown of glycogen to glucose. And you need to be able to talk your way through that series of reactions. So how is the G protein activated exactly? So what happens is if you've got an inactivated G protein, the G protein has actually two specific areas. There's an alpha site and a beta site to it, and actually, a gamma site here as well. So you've got alpha, beta, and gamma sites of the G protein. So when it is inactivated, there is a molecule called GDP or guanosine diphosphate that is bounded to the alpha subunit, as you can see here. When the protein becomes activated, the ligand attaches to the receptor protein on the membrane. This then ligand attaching causes the alpha molecule of the G protein to change shape, and that releases the GDP molecule out of the alpha site. That then allows GTP, which is guanidine triphosphate, to attach to the G protein, and that causes it to then, the G protein, as you see here, to separate into its subunits, which then causes further changes into the cell. This triggers a response in the cell by the ligand, so that'll probably activate enzymes. So the key thing here is when it's not being activated, the G protein and its three alpha, beta, and gamma subunits are stuck together with GDP. The GDP is released when the ligand attaches to the receptor protein. That then allows GTP to attach to the alpha unit, and the alpha unit then separates it from its other subunits, and that triggers the further changes in the cell. Now, one example that you need to know is something called a kinase enzyme. So kinase enzymes are ones that phosphorylase molecules using ATP. So we just mentioned it in the adrenaline series of reactions. Insulin receptor is one that is affected by this. Insulin receptor is made of two tails. You can see them here. Okay, when insulin binds, the tails form together, so they go from being separate together, forming something called a dimer. And each tail phosphorylates the other one, so it exchanges phosphorus, and that causes a series of biochemical reactions that then trigger glucose transporters to move to the membrane and enter the cell. So when the insulin is activated, when the enzymes are activated by insulin attaching to it, that causes a series of reactions that causes these vesicles with these GLUT4 proteins to attach to the receptor, and then glucose can enter the liver cell, and that can be stored away as glycogen to lower your blood sugar levels.